This set of slides will cover immunology and epidemiology, the freedom from disease, how we track disease, who gets sick, and why. The balancing act between pathogens and us is often determined by the health of our bodies. Our health is largely supported by our immune system. There are innate factors, or what I often describe as nonspecific factors, that also protect us from infectious diseases, which are still a problem today. The combination of an active immune system, as well as various physical and chemical barriers, tilt this balancing act into our favor. But with this illustration, can we really determine which one is the pathogen? Unfortunately, oftentimes the pathogens do succeed in tilting that balancing act into their favor. Pathogens produce chemicals or factors called virulence factors. These virulence factors, both found as the somatic structure or integral parts of their cells, such as the Pseudomonas aeruginosa example with LPS or lipopolysaccharide, which are considered endotoxins or within the part of the cell, as well as excreted products or exotoxins, such as tetanus toxin and diphtheria toxin. All toxins are biological poisons, whether they are endotoxins, part of the structure of the cell, or exotoxins, which are excreted products, chemicals that are produced inside the cells of the bacteria that are then transported out into our tissues and body organs. All of these virulence factors can have quite deleterious or pathological effects on our cells. That's the essentially function of these various virulence factors, to provide opportunities or means by which the pathogen can succeed in growing and reproducing, and thus, of course, causing our infections. An example of essentially a somatic or integral type of virulence factor would be seen in Listeria monocytogenes. Here we have an obligate intracellular pathogen, which means that this particular pathogen must gain entry into our cells to start infection. Listeria gains entry into our cells by passing through our cytoplasmic membranes. Once inside, there's a structure on the tail end of these rod-shaped Listeria cells. It's called Act A. Act A is a single virulence factor that has the ability to collect actin filaments, which are found in eukaryotic cells. Actin filaments in eukaryotic cells function very much like rubber bands. They're very stretchy filaments that our cells use as part of our cytoskeletal structure. But what Listeria can do, using Act A almost as a hook, it gathers up actin filaments inside of our cells, assembles them into what's described as a comet tail. It will use these actin filaments to propel the rod-shaped Listerial cells within our cells, essentially searching for food and moving around, and these comet tails also will propel these cells of Listeria between our cells, particularly within tissues, which allows Listeria to remain hidden or unavailable to our own immune system factors, which try to target and eliminate any type of pathogenic organism. So Listeria is a very successful intracellular pathogen. You may hear of Listeria infections, often associated with food. There are different types of foods especially dairy products, processed meats, which might possibly be sources of listeria, having been contaminated in the manufacturing process. Listeria on the move. Here are two photos or illustrations of a magnified image of a single cell of listeria with those actin filaments forming the comet tail on the left. And then on the right-hand side, with what was essentially a confocal or contrast imaging microscope, you can see these listerial cells escaping from an infected eukaryotic cell, again, using the comet tails to propel the individual bacterial cells looking and searching for a new eukaryotic host cell to infect. Nonspecific and specific factors in our body provide us protection or freedom from disease. This freedom from disease is generally described as immunity. Some textbooks and some immunologists refer to the phrase innate and adaptive. Innate is analogous to what I describe as nonspecific, and the adaptive would be the specific. I find that students get a little confused with those words innate and adaptive, particularly the way they're defined in most or many textbooks. There are still textbooks, including ones that I prefer, that use phrases like nonspecific and specific, which I find students are able to more clearly define or separate in their minds in remembering which type of protective device or which type of protective system we're talking about, whether it's nonspecific or specific. Nonspecific immunity are the first defenses that protect us from invasion by potentially pathogenic microorganisms. The first defenses can be simply physical barriers, intact skin and mucous membrane. As long as those cells lining the skin and mucous membranes are intact, that prevents the introduction of potentially pathogenic organisms. We have chemicals on those various body surfaces that also provide us protection. The fatty acids, which provide a particular pH, the different types of simple biochemicals often provide a chemical barrier. We have cells and we have body fluids. Tears, for example. Simply that fluid bathing our eye, the water and the chemicals in those cells and in those fluids are going to give us various types of nonspecific first-line defense. Specific immunity, on the other hand, is referring to a particular type of body system. In anatomy and physiology, we talk about body systems, like the respiratory system, the musculoskeletal system. We also have an immune system that's comprised of various organized tissues and organs with unique types of cells in that immune system that confer what we call specific immunity. We'll describe those in more detail in future slides. One of the earliest signs that something's wrong is the appearance of symptoms and signs of inflammation. Inflammation is an activation of various nonspecific factors that provide early first-line defense. The inflammatory process is often due to trauma, where we've been injured, infection, unwanted growth of pathogenic or potentially pathogenic organisms, or even abnormal cells in the form of tumors. These are all possible triggers that would activate the inflammatory process. Inflammation was once thought to be unimportant, that it was simply the visible signs of something's going wrong, but that it really didn't have any connection to what happened later in an infection or disease. Well, we know now that that's not true. The events that occur early in the stages of an inflammatory process often determine or contribute to the outcome, the success or failure of our various nonspecific and specific immune factors to eliminate or cure us of that particular disease or infection. So the interactions between the nonspecific immune factors that are our first-line early defense and the pathogen and the tumor often communicate or lead to activation of more elegant and more powerful specific immune factors that are then also largely responsible for cleaning up or ending the infection or disease. The symptoms of inflammation are edema, the accumulation of body fluid, erythema, redness, dysesthesia, pain, and heat, an increase in localized temperature. The inflammatory process can be triggered by simply a breach in the physical barrier, such as a cut that allows bacteria to get beneath the surface of the skin. There are unique biochemical features on the surface of the bacteria that interact with the damaged cells within our skin. The release of various types of vasoactive amines, like histamine and bradykinin, trigger further reaction of blood vessels and pain receptors. Capillaries dilate, vasodilation, which brings more blood to the tissues. The idea, of course, the reaction, is to bring more blood and oxygenation to the tissues for healing and the white blood cells that are going to be part of the cleanup process. As a result of that vasodilation, though, the skin becomes reddened or erythematous and warmer. We see that increase in temperature. As the capillaries continue to react to the biochemical release factors from the pathogenic organisms or potentially pathogenic, as well as the cells, including the white blood cells that have been recruited to the area, the capillaries of blood vessels in that skin become more permeable. Fluid leaks out of the blood vessel into the tissues, as well as various white blood cells migrate from the bloodstream into the tissues to attack and eliminate pathogenic organisms. So we see swelling as the cells move into the affected tissues. Blood clotting occurs. As we see blood entering into the wound, it will fill the space or the opening and, of course, form that gel-like blood that prevents any further bleeding. As that blood clot forms, it eventually dries into a scab. This provides a seal and prevents any other introduction of unwanted bacteria. But what if the bacteria continue to grow inside the cut, underneath the scab, and in the skin? That's where the white blood cells that are responsible for the cleanup enter into this area, transferring from the blood vessels into the tissues. The cells called phagocytes gobble up or phagocytose the bacteria. They're going to move through the blood vessels by a process called diapedesis. So the movement of phagocytic white blood cells from the blood into infected tissue is called diapedesis. These phagocytes are attracted to the bacteria and any dead and dying cells of our skin, and they're going to migrate by attraction to these chemicals. The attraction to various chemicals that triggers movement of cells is called chemotaxis. And that's a general term that could be applied to any kind of cell that's attracted and moves towards a chemical. This chemotaxis is a general term that could be applied to bacteria as well as eukaryotic cells moving toward a chemical attractant. And that's what these phagocytic cells will do. They're attracted to various chemicals released by this destructive process between the bacteria that are growing and the cells of our skin that are being destroyed or damaged. Their larger blood vessels continue to dilate in response to, again, the chemicals that are circulating in this localized area. And then if you look, the visual image of this infected wound, small or large, you'll start to see that it swells. You might see some discoloration because of the erythema, so it's becoming very red, maybe hot, swollen. And you might even see the accumulation of the battle, if you will, between the bacteria and the phagocytes in the form of a sort of grayish, greenish, yellowish, gooey substance that we generally describe as pus. That's where we have that battle between our phagocytic white blood cells and the growing bacteria. As dead and dying cells of both bacteria and our skin cells collect and accumulate, other types of body fluids and phagocytes remove all of this debris. Eventually, the various types of cells in the skin begin to grow and proliferate and form this, essentially the scar. The scab falls off, and the connective tissue in the cells that have now replaced the damaged area have now sealed up and formed a nice, intact, complete physical barrier once again. So that inflammatory process is very important to initiate and to be part of the healing process of eliminating even a simple wound. A primary source of white blood cells for our body and our immune defense is the bone marrow found in areas such as the long bones of the femur, the iliac crest of the pelvis, and even in younger individuals in the sternum. Initially, those cells in the bone marrow that are responsible for the production of all types of white blood cells are called hematopoietic, pluripotent stem cells. Pluripotent means these cells have potential to transform or change into virtually any type of cell. And then hematopoietic is a descriptive that tells us what kind or type of cells. Blood cells. Not only white blood cells, but also red blood cells, and even tiny little pieces of cytoplasmic membrane and cytoplasmic contents of cells that we call platelets. Well, they're not really cells, they're fragments of cells, but that's for further discussion. Pluripotent hematopoietic stem cells go through transformations based on chemical signals of interleukins and various other kinds of cytokines. These chemical signals essentially help to regulate or change genes inside of the stem cell to begin the transformation into either erythrocytes, which are red blood cells, platelets, which are fragments of cytoplasmic membrane and cytoplasm from megakaryocytes in the bone marrow, various types of granulocytes produced in bone marrow, such as basophils, eosinophils, neutrophils. There are other kinds of cells like monocytes, agranulocytic cells such as TMB lymphocytes, and even natural killer cells. All of these types of red and white blood cells are derived initially from the same progenitor cell, hematopoietic stem cell, in bone marrow. Neutrophils are a type of white blood cell, again, originally produced from bone marrow, that wind up in blood circulation and body tissues. These are the primary phagocytes of the human body. Phagocytes are the cells that ingest or phagocytize various types of particles, bacteria, dead and dying cells within our own body. And the illustration or the typical description of the process of phagocytosis begins with the formation of essentially a vesicle or a phagosome, as the bacteria are brought inside of the neutrophil by a type of specialized endocytosis. The extension of the cytoplasmic membrane of the white blood cell surrounds the bacteria, draws the bacteria in, now surrounded with this membrane-bound structure we call a phagosome. A phagosome, once inside the neutrophil, fuses with some of the granular contents of the neutrophil that are lysosomes. Oftentimes a generalized definition or description of a lysosome is it's like a mini stomach within the cell. It's not really a stomach, but the lysosomes contain either precursors or completely formed proteolytic enzymes, in addition to enzymes that can break down proteins, the proteolytic enzymes. We also have enzymes that can break down lipids and enzymes that can break down carbohydrates. So all types of digestive enzymes can be found within these various liposomes, which are seen as granules through the microscope in the cytoplasm of a neutrophil. As the lysosomes fuse with the phagosomes, we now have a newly formed structure called a phagolysosome. The digestive enzyme contents of the lysosome are dumped into the phagosome, and we see the breakdown or digestion of the bacterial cell. The residual, what I call dead body parts of bacteria post-digestion, are then exocytosed or evacuated from the neutrophil. Eventually, depending upon how many bacterial cells are ingested and broken down, the neutrophil essentially is completely exhausted. It has no further granule contents to provide any defense, and the cell, the neutrophil itself, dies after it's gone through a series of battles, if you will, or ingestions of various bacterial cells. Other types of non-specific white blood cells that provide a type of defense or first-line immunity are the eosinophils and the basophils. These too are granulocytes. Collectively, the neutrophils, eosinophils, and basophils are described as granulocytes because we see granules in their cytoplasm. The eosinophils are responsible for protecting us from parasites, such as worms or different types of animals that might infect our bodies. And also, eosinophils are responsible for the symptoms that we associate with different types of allergies. Bee sting allergies, penicillin, shellfish allergies. These are often triggered by different types of vasoactive amines released by activated eosinophils. Basophils are also a type of granulocyte that are responsible also for some of the allergic symptoms that we see in, again, pollen allergies, ragweed allergies, penicillin allergies. Different kinds of allergic reactions often have both eosinophils and basophils initiating or triggering those symptoms that we associate with various allergies. If the nonspecific first-line defenses aren't successful at preventing the continued growth of unwanted, potentially pathogenic bacteria, then the specific factors must be recruited. And in fact, that's what the nonspecific cells and chemicals do. They continually release, essentially, alerts to recruit the nonspecific lymphocytic types of reactions or specific cells. And those specific factors then, after 3 or 4 days, can start to enter into the battle. These are the heavy hitters, if you will, the cells and the chemicals that should be able to surmount a powerful reaction and reverse the effect or the infection or disease. Oftentimes, though, we have to distinguish who is who. Which cells are infected in our bodies, which cells are us and not us. To do that, we have particular types of structures on our own individual cells in our bodies. These cells are labeled and described as major histocompatibility complex markers. A sort of long phrase that can simply be described as self-markers. If you've ever heard of someone going for an organ transplant and they have to go for tissue typing or tissue matching, these are the structures on the donor organs that are matched up or correlated with the same types of self or tissue markers on the recipient. Ideally, we want a pretty good match. We want that organ MHC, or major histocompatibility complex markers, on the donor tissue to match up pretty closely with the recipient's MHC. If there's not a close match, then there's a good chance that the specific factors that are part of the immune system might erroneously recognize the donor organ as non-self, something that's not supposed to be there, and the specific factors will react accordingly, and in fact attack that type of donor tissue as it would attack or try to eliminate an unwanted growth of either tumor cells or potentially pathogenic bacterial cells. So these self-markers are vitally important to distinguish self from non-self, and they're found on the surface, embedded in the cytoplasmic membrane of all cells in our body that have nuclei. Cells that communicate between the nonspecific and specific factors of immunity are called antigen-presenting cells, or APCs. Antigen-presenting cells wind up distributed throughout the body, but they can concentrate within specific areas in the organs or organized tissues of the formal immune system. Some of the organized tissues or organs associated with the formal, proper type of immune system that's recognized anatomically are lymph nodes and spleen and thymus. The lymph nodes in particular are areas where APCs, or antigen-presenting cells, reside. We have 3 different categories or types of antigen-presenting cells, APCs, that can communicate between the nonspecific early first response when a bacterial pathogen is trying to invade, and the specific factors, which are much more powerful and more capable of addressing or eliminating unwanted infection. We have dendritic cells that are found just underneath the surface of our skin, the mucous membranes. These can function as APCs, transporting viral antigens from mucous membranes and again skin to various areas of the lymph node where they can communicate with lymphocytes found also in the lymph node in the areas pretty much throughout the body, the cortex, and the medulla of the lymph node. we have macrophages that are responsible for phagocytizing bacteria. And we've already mentioned neutrophils as the primary phagocyte found in the human body. But macrophages are also phagocytes, but they have a special function. Unlike neutrophils that really aren't capable of communicating or transferring information to the specific arm of immunity, the macrophages are able to do this. They're essentially the bridge or the link between non-specific phagocytes and the lymphocytes responsible for eventually producing a very powerful immune response. Macrophages can be found throughout the entire lymph node, in the medulla, the cortex, anywhere else, the follicles. That's where we can find the macrophages that have essentially ingested unwanted bacteria. B cells, B lymphocytes, can function as APCs. Oftentimes this surprises students because usually they get the idea that APCs are usually always all non-specific cells, but they're not. B lymphocytes capture microbial toxins. They localize in specialized areas illustrated as follicles or germinal centers. Once located in those germinal centers, the B lymphocytes, in fact, communicate with other types of lymphocytes called T lymphocytes, again, communicating or transferring that information to the T cells in the lymph node. APCs undergo what we call processing and presentation of bits and pieces of other viruses, bacteria, or toxins that we call antigens to T cells via what's called the MHC, or the self-marker. The processing is the breaking down of the virus, bacteria, or the toxin. The presentation is packaging bits and pieces of those items into self-markers and essentially displaying or showing the T cells what has been captured in the self-marker. APCs are the link between nonspecific and specific immunity. T lymphocytes will only recognize non-self items when presented, those non-self items, in a self-marker by an APC. It's what we call MHC restriction. Lymphocytes can only essentially detect the presence of viruses, tumors, bacteria, or toxins when bits and pieces of those items are packaged in a self-marker and displayed to the lymphocyte, the T lymphocyte, directly. Different pathogens and different items like toxins are processed slightly differently, but they're all packaged in self-markers for presentation to the T lymphocytes. The types of cells, T cells, that are responsible for reacting and recognizing antigen or non-self, initially starts out as an immature T lymphocyte released from the bone marrow. Those immature T lymphocytes have to be processed or educated in the thymus. Once emerging from the thymus, they have differentiated into specialized T cells, of which there's a vast network of various types of T lymphocytes. Some general categories of T lymphocytes that have been thoroughly investigated and delineated or determined to exist are the T helper cells, the T cytotoxic cells, and the memory T cells. T helper cells have been identified as carrying a structure on their cytoplasmic membrane that's designated CD4 positive. For cytotoxic T cells, those have been identified as carrying a marker that's been identified or designated as CD8 positive cells. And so the language of an immunologist is often like alphabet soup. They talk about T helper CD4s, T cytotoxic CD8s, And lots of acronyms or abbreviations pepper any discussion or research paper involving specific factors of the immune system. Now, these are not the only types of T lymphocytes. Remember, there's a huge vast network that's been discovered of very specialized and very important T lymphocytes. An example of the function of CD8 positive T cytotoxic cells is when a macrophage or some other APC ingests a toxin, bacterium, or virus. That toxin, bacteria, or virus is then processed or digested or broken down, and fragments of those bits and pieces of materials are packaged in a self-marker or MHC class 1 marker. We identified two different categories of MHC markers, class 1 and 2. And to activate a CD8 positive T cytotoxic cell, fragments of antigen, again, these are bits and pieces of toxins, bacteria, or viruses, are packaged in this class 1 marker. If we want to communicate with different types of T lymphocytes, we're going to package these bits and pieces into a different type of MHC marker. So to communicate with the CD8 cytotoxic cells, we have to package the antigen in a class 1 MHC. These sort of pre-T cells, CD8 positive, interact with the fragment or the antigen in the MHC class 1 marker and get alerted by direct contact. So these cells really do lock together briefly, and there's almost as if these cells are detecting sort of different chemical signals with a lock and key, or what's called an induced fit mechanism. They interact with one another directly. The information is essentially chemically transferred from one cell to the next, and this CD8 T cytotoxic cell now becomes activated. Once the CD8 cell becomes activated through this induced fit mechanism, then the CD8 positive cell releases destructive enzymes that can then kill the abnormal or infected cell that contains the real virus or bacteria or toxin. So the T cytotoxic cell really is a killer cell. It's the cell that goes around destroying virally infected or infected cells that are being affected by intracellular bacteria in your body. CD4 positive cells become activated in a little bit more complex way. And in fact, naive CD4 positive T lymphocytes, once activated, can become further differentiated into other subpopulations that we call T helper 1s and T helper 2s, Th1s and Th2s. We have a similar type of antigen presentation that we've described before, where we have bits and pieces of toxins, bacteria, or viruses that are packaged in self-markers that then come in direct contact with the CD4 positive T cell to activate that cell and initiate through cytokines and interleukins further differentiation into those Th1 or Th2 subsets. Notice that in addition to creating active functioning CD4 Th1s and Th2s, we also generate a population of memory CD4 positive cells. This is why vaccines work. In a vaccine, particularly with a booster shot of a vaccine, we're eliciting and activating that memory, that pool of memory lymphocytes that can remember or more quickly respond to the same antigen that was initially used in the first vaccine exposure. The type of MHC marker that's used to present antigen to CD4 positive cells is called a class 2 MHC marker. It's still a self marker. It's just a slightly different version called class 2. Again, a bitter piece of the pathogen or toxin is packaged in the class 2 self marker, and that's directly in touch with some structures that are found that are complementary to the MHC antigen complex. And we have, again, by induced fit, an interaction between the T lymphocyte and the APC, presenting this structure to the CD4 positive lymphocyte. The CD4 positive lymphocyte transforms into either Th1. In that case, the Th1 cells are responsible for continuing to promote the function of destroying any kind of intracellular pathogen and conferring what's called cell-mediated immunity to intracellular pathogens. If instead we have cytokine or interleukin activation that promotes the growth of Th2 CD4 positive cells, those types of T cells are responsible for enhancing the function of B lymphocytes and eliminating extracellular materials such as toxins and extracellular bacteria using chemicals called antibodies produced by B lymphocytes. This is called humoral immunity, where we're eliciting the major function of B cells in the production of antibodies using T help from the Th2s. I know it gets very confusing and complicated, and in fact, this portion of the descriptions of immunity through the proper function of specific effectors of the immune system is very complicated. It's taken scientists many years to dissect or figure out this very complex network of cells and the intricate balance between the function of the lymphocytes and the eventual elimination of either tumors, pathogens, or any other unwanted type of factor that's affecting our bodies. So don't worry if it's a little bit complicated or confusing. Don't hesitate to ask if you have any questions, and let me know if I can help you in any way.